Hi, Lori. Happy New Year 2021. You're looking good on Zoom today.
Likewise, David. Would you like to take a little walk down memory lane explaining to people how our self-repression collaboration came about?
Sure thing. That's one good thing to come out of our virtual lifestyle this past year. It's been a while since we spoke in person at Wayne State after my visit last February.
Yes, we heard there about your work on the retinoblastoma tumor suppressor and how the fly RBF2 protein is on the promoter of so many widely active genes, including those for the ribosomal proteins. And rather than shutting them down entirely, the corepressors seem to only dampen the activity.
Right, that's what made me think about your discussion of the SIN3 co-repressor when you spoke here at Michigan State. Regulation of metabolically important genes, but not a decisive on-off switch, enough to control metabolism of methionine in an important way.
Yeah, somehow the field of transcriptional repression has gotten stuck on the notion of repressors as a lockdown mechanism. And for cell type specific expression of genes, that certainly seems to be the case.
Exactly. I cut my teeth on stripe enhancers from the fly embryo, and their repressors like KNRPs and Kripl do exactly that.
And not to mention the cell cycle regulation by retinoblastoma proteins. Those genes do seem to need some rather complete shutoff mechanisms. Anyway, repressors do silence genes, but we find that for SIN3, they also have a crucial role in modulating gene activity without the absolute shutoff that's needed for those highly cell type specific genes.
Can you explain exactly what you're thinking about with the term soft repression in this context?
Sure, let me just share this cartoon with you. All right, so we have for many genes an enhancer that is bound by an activator, and that leads to recruitment of the basal transcription machinery. Transcripts are produced by RNA polymerase II, and off we go.
Awesome graphics, Lori.
Well, that is thanks to our collaborator, Anna Maria. So now let's consider the classic repressor in mechanism. So we have a gene and it's humming along.
Now let me guess what's going to happen next. The repressor binds, and through chromatin-based mechanisms, the gene is silenced. Hard repression.
Exactly. So now let's consider what might happen for something of a soft repressor. Here is really the key point. There are potentially thousands of targets of SIN3 and Rb, and many of these are genes involved in metabolic regulation. And you don't want them to go from 90 miles an hour to 0 in an instant. We know in the Motor City, that's a recipe for disaster.
Right, going from 90 miles to 70 miles an hour might be the prudent thing to do. For one, it may get you out of a speeding ticket.
Sure. And for many metabolic pathways, this is the kind of regulation that makes sense. Here, let's see how this would play out. We have a gene and it's humming along, but we need a little less output due to some signaling. And so here comes our co-repressor, and it associates with the promoter, and it attenuates the transcription. And it does not cancel the output, but lowers the transcriptional output.
So that is soft repression.
Exactly.
So why the neologism of soft repression? I haven't had one of those since the days of billboard enhancer.
Well, it's important to point out this type of regulation because we've been missing out on a large part of cellular regulation. SIN3 appears to be dedicated almost entirely to this sort of activity. If we use stringent cutoffs to our transcriptomic data analysis, you might not even understand how this evolutionarily conserved essential regulator does its job. For quantitative understanding of regulatory networks and for interpreting genomic data, we should consider this possibility that soft repression is a major part of how eukaryotic cells are wired.
And it may not only be SIN3 and Rb. From genomic surveys, we know quite a few transcriptional regulators that are parked around the genome, yet only a small percentage of genes are typically shown to be directly regulated.
And sure, there still might be off-target or biologically irrelevant interactions, of course. All that glisters is not gold.
You mean glitters?
I was quoting the Bard lad. Just brush up on your Shakespeare and we'll take this discussion to a higher level.
Anyway, check out our new paper in BioEssays for why transcription development and disease researchers should think about soft repression. It might open some new horizons in your own work.
I couldn't have said it better. And of course, thanks to Anindita, Anna Maria, and Stephanie for pulling this collaboration together. And of course, our funding agencies for their support.
From the capital city to the Motor City, signing off for now. Until soon.